My name is Christine Boutros. I am a six-year MD PhD candidate at UCSF and a fourth-year neuroscience PhD candidate. The project that ARCS has so kindly funded for the last 2 years is my thesis project on valley fever meningitis. So valley fever is a fungal disease that is endemic to the southwestern United States. And in a small percentage of patients who get this disease, it spreads to the brain, causing life-altering meningitis and lifelong fungal infection. One of the things that is important about fungal diseases is that there are no known fungal vaccines for prevention, and yet 10 to 15% of the fungi that we know about, of which there are hundreds of thousands, 10 to 15% of them are capable of neuroinfection. It's really important for us to be able to surveil accurately and diagnose people early so that we can make sure that folks get appropriate care and get ahead of this disease to relieve disease burden. With patients who have valley fever meningitis, folks have to undergo lifelong antifungal therapies, and they're never fully cured of the disease. So it's a high concern for us that we better treat these patients. So with that in mind, valley fever meningitis has a diagnostic problem that my thesis hopes to address. One of the things is that there's very little actual fungus in the brain, and so we can't go ahead and directly detect the fungus. Usually the meningitis itself, which is inflammation of the lining of the brain, that inflammation is more of a result of our host immune system, the human immune system, responding to the fungus. So it can be quite difficult to actually detect the fungus itself. Another issue we have is that the immune system is kind of compartmentalized in the brain because of the blood-brain barrier. So things that are happening in the rest of your body are not necessarily happening in the brain, and vice versa. So that poses an issue because a lot of the targets that we've identified have come from sampling of the blood from what we call the periphery. And those targets that were detected in the periphery are not necessarily the same targets that are present in the brain. So with all those considerations in mind, I've been able to develop a new way of profiling the human antibody response in the fluid that surrounds the brain. That's called the cerebrospinal fluid. And I express all these different proteins that come from the causative fungus of valley fever on the surface of these small molecules and figure out what antibodies are being produced in the humans against those proteins. So on the bottom here, you will see what we call a heat map. And there's a very clear and very different profile in valley fever meningitis cases than in the controls. And these controls are people who have other neuroinflammatory disease. And these are CSF that were sampled. So it's really clear that there's a very unique profile in cases that we don't see in controls. And these are now new candidates that we could use to improve our current diagnostics. On the right is an example of a diagnostic that we're trying to build in the lab with the information that was gained from that original phage immunoprecipitation and sequencing assay that I was showing on the bottom here. So in this graph on the right, we've been able to identify a antigenic protein that is very, very differently detected by the immune system, so much so that in patients who do not have infection, there is almost no response. That's why all those gray dots are on the bottom. And on the left, in blue, are the CSF from people with valley fever meningitis, and there's a huge range of detection. So we really think that this approach is going to open the doors for modern, up-to-date, and improved diagnostics in the valley fever field. Thank you.